The PLE-REAP application and all of its contents as well as the flipbooks are copyrighted by the Intellectual Property Office of the Philippines, thus subject to copyright laws. Reproduction, by any means, of the material and or its parts is punishable under the full extent of the law.
Okay, so let's continue now. This time we shall be discussing cellular respiration. So this constitutes glycolysis, Krebs cycle, and the electron transport chain. Right? So the first part that we shall be discussing regarding cellular respiration is glycolysis. So what is glycolysis? So this is the process by which chemical energy or food is broken down and the energy is released. And of course, that energy is partially captured into ATP, partially captured because the other energies are released in the form of heat. And as we know, we can use carbohydrate, fat, and protein as fuel, but of course, glucose is the most common example in which these pathways are involved. So cellular respiration can take place in the cytosol and in the mitochondria. Now, the process where cellular respiration takes place can be divided into three major pathways. In glycolysis, there is in the cytosol, as you can see here, takes place in the cytosol. A continuation of cytosol is the Krebs cycle that occurs within the mitochondrial matrix. And in the inner mitochondrial membrane, there is oxidative phosphorylation via the electron transport chain. Now, these three processes are only possible, the complete three processes are only possible in the presence of oxygen. What about if oxygen is absent? In the absence of oxygen, the process stops at glycolysis. So there is no Krebs and no oxidative phosphorylation. And there are two metabolic pathways that are possible in the absence of oxygen. That would be glycolysis and fermentation, as we shall see later on. So what is this glycolysis? This is a process where a 6-carbon sugar is oxidized and broken down into two 3-carbon molecule called pyruvate. So as you can see from this illustration, glucose is broken down into 2 pyruvate. Now the process results is accompanied by a gain of 2 ATP and 2 NADH molecule, which can be later converted into ATP. Now, why do we gain 2 ATP? Well, initially there is an investment of 2 ATP, and we gain 4 ATPs near the end. So 4 minus 2 would be 2, so there's a net gain of only 2 ATP. So pyruvate can be converted to acetyl-CoA and can enter the Krebs, but only in the presence of oxygen. So the breakdown of glucose into 2 pyruvate molecules as shown by this reaction to release energy is called glycolysis. It's a catabolic pathway that can be either aerobic or anaerobic. So either way, it is possible, and it occurs in the cytosol. We can divide glycolysis into 3 major parts. Let's divide it into 3 major parts. So the first process is that glucose is phosphorylated using ATP as the source of phosphate into glucose 6-phosphate. This is catalyzed by glucokinase in the liver or hexokinase in all other organs. Glucose 6-phosphate is converted into fructose, not shown here, and then phosphorylated for the second time using ATP as the source of phosphate to yield fructose 1,6-biphosphate. Fructose 1,6-biphosphate is split into two components. One is glyceraldehyde 3-phosphate. The other component is dihydroxyacetone. Glyceraldehyde 3-phosphate can proceed right away to the next phase, but dihydroxyacetone has to be converted into glyceraldehyde 3-phosphate. So from here on, there are now two glyceraldehyde 3-phosphate. It will be oxidized to yield 1,3-biphosphoglycerate, and in the process, NADH is generated. Since there are two phosphoglycerate, there are two NADH that will be generated. Then it will undergo substrate level phosphorylation, wherein ATP is produced. Since there are two 1,3-biphosphoglycerate, 2 ATP will be produced. 3-phosphoglycerate is converted into phosphoenol pyruvate, and the final step is another substrate level phosphorylation, wherein the two phosphoenol pyruvate are converted into pyruvic acid. This generates 2 ATP. So we can divide glycolysis into 3 stages. The first 2 phosphorylation process is the energy investing stage. When we split fructose 1,6-biphosphate into 2 components, this is the lysis stage. And the final stages where we generate NADH and ATP via substrate level phosphorylation, this is referred to as the energy harvesting stage. So what are the important steps in glycolysis? The first is the conversion of glucose to glucose 1-phosphate. This is catalyzed by hexokinase or glucokinase in the liver. This results in the expenditure of ATP, followed by converting glucose phosphate into fructose, after which it is phosphorylated for the second time through phosphofructokinase. This requires expenditure of ATP. Now take note, hexokinase and phosphofructokinase are 2 of the 3 important enzymes in glycolysis. Then this will be split into 2. One component is dihydroxyacetone, which must be converted into glyceraldehyde 3-phosphate. The other component is glyceraldehyde 3-phosphate right away. So now there are 2 glyceraldehyde 3-phosphate. This will undergo the final stage wherein there is generation of NADH catalyzed by glyceraldehyde 3-phosphate dehydrogenase, substrate level phosphorylation that produces ATP catalyzed by phosphoglycerate kinase, and the final substrate level phosphorylation that generates ATP catalyzed by pyruvate kinase. Right, so what are the 2 enzymes involved in substrate level phosphorylation? Phosphoglycerate kinase and pyruvate kinase. And the step that produces NADH would be glyceraldehyde 3-phosphate dehydrogenase. Okay, so the overall reaction is shown here. Glucose is going to be phosphorylated twice. This is the energy investing stage that generates phosphofructokinase that is split into 2. Glyceraldehyde 3-phosphate and phosphoglycerate kinase, phosphoglycerate, each of which will generate NADH, ATP via substrate level phosphorylation, and the final product is pyruvate. So there is 4 ATPs generated, but we invested 2, so the net gain would be 2 ATP. What will happen now to the pyruvate? That would depend on the presence or absence of oxygen and the type of organism that is involved. In all organisms, in the presence of oxygen, pyruvate is decarboxylate, enters the mitochondria, and the pyruvate is decarboxylated to yield acetyl-CoA. And the enzyme here is pyruvate dehydrogenase. This acetyl-CoA, as you know, may now enter the Krebs cycle, but this is only possible in the presence of oxygen. But what if oxygen is absent? Let's take a look at what happens in higher organism. Pyruvate does not enter the mitochondria. It remains in the cytosol and is converted into lactate by the enzyme lactate dehydrogenase. This is to ensure that glycolysis continues to run despite the absence of oxygen. Lactate is what causes soreness of muscle. What about in lower organisms like yeast? What would happen now to pyruvate if there is no oxygen? In yeast, it's a different pathway. Pyruvate is converted into ethanol by pyruvate decarboxylase. So alcohol. Pag walang oxygen sa lower organisms like yeast. So in other words, there are three fates for pyruvate. Across all organisms in the presence of oxygen, pyruvate enters the mitochondria and is converted to acetyl-CoA by pyruvate dehydrogenase. In higher organisms like humans, pag walang oxygen, pyruvate remains in the cytosol converted into lactate dehydrogenase. But in lower organisms like yeast, pyruvate is converted into ethanol. So just to recap, what happens to pyruvate? The answer to that is it depends on the organism and the condition. Let's start with lower organisms like yeast. Under anaerobic condition, pyruvate is acted upon by the enzyme pyruvate decarboxylase, converting it into ethanol. But under aerobic condition, pyruvate enters the mitochondria and is converted into acetyl-CoA that can enter the Krebs cycle. Higher organisms, others, tayo yun. Under anaerobic condition, pyruvate is acted upon by lactate dehydrogenase and is converted into lactate. And this is what makes the muscle sore. Now, this ensures that the glycolytic pathway continues to run even under the absence of oxygen. But if oxygen is present, just the same, pyruvate enters the mitochondria and is converted into acetyl-CoA. So what are the key points you need to remember regarding glycolysis? So what is glycolysis? This is the process by which glucose is a 6-carbon sugar is oxidized, broken down into 3-carbon product. The end product is called pyruvate. Under what conditions can glycolysis occur? It can occur either in aerobic or anaerobic condition in the cytosol. What about for aerobic condition? What would be the yield? 2 ATP and 2 NADH. How do we regulate glycolysis? Well, there are 3 important enzymes you need to remember regarding glycolysis. The regulatory enzymes are hexokinase, otherwise known as glycokinase in the liver, phosphofructokinase, and finally, pyruvate kinase. These enzymes catalyze the irreversible steps in glycolysis. So just remember this because later on in gluconeogenesis, which is essentially the reversal of glycolysis, these steps cannot be reversed, and therefore there has to be bypass step catalyzed by a different set of enzymes in gluconeogenesis. So again, what are the 3 important enzymes and steps? Phosphorylation of glucose catalyzed by hexokinase or glucokinase. Phosphorylation of fructose 6-phosphate catalyzed by phosphofructokinase. And the final step is catalyzed by pyruvate kinase that involves the formation of pyruvate. So let's continue with what happens to pyruvate under aerobic condition. And this, of course, occurs now in the mitochondrial matrix the process is what we call Krebs cycle. So let's continue now with cellular respiration. And the second part here is the Krebs cycle. Where does it occur? Of course, it occurs in the mitochondrial matrix. And the purpose of the Krebs cycle is to generate reducing equivalent from NADH and FADH2, as well as indirectly generate ATP. Now, pyruvate that comes from the glycolytic pathway, of course, has to be converted into acetyl-CoA. This is a 2-carbon molecule. Now, when acetyl-CoA is incorporated into the Krebs cycle and it is oxidized, the chemical energy is released and captured in the form of reducing equivalents, NADH, FADH2, and ATP. What is the significance of Krebs cycle? It is very important because it is the main source of energy for cells. As a matter of fact, the important cells in your body, like the neurons, the muscle, the cardiac muscle, all depend on the Krebs cycle as their energy source. This is an important part of aerobic respiration. So the mitochondria harnesses the chemical energy of acetyl-CoA and captures it in the form of reducing power, NADH, FADH2, as well as GTP. That will be converted into ATP later on. So the goal of Krebs cycle is to generate ATP by generating NADH and FADH2. Now, for ATP to run, it requires some carbohydrate. So where does acetyl-CoA come from? Like what we have said earlier, pyruvate from glycolysis enters the mitochondria and is converted into acetyl-CoA. This already generates the first NADH, right? So there are 2 acetyl-CoA coming from glucose. And the process of converting pyruvate to acetyl-CoA is called oxidative decarboxylation that also generates NADH. Now, what will be the fate of acetyl-CoA? So we did say that acetyl-CoA enters the Krebs, right? But that's not the only fate of acetyl-CoA. Depende pa yan sa metabolic state ng cell. So one fate of acetyl-CoA is that it can enter the Krebs cycle. But this only occurs under the following condition. In the presence of carbon dioxide under aerobic conditions. So acetyl-CoA is metabolized to carbon dioxide, NADH, FADH2, GTP, and ultimately ATP. Now, what if you already have a lot of energy? You don't need extra energy. What happens to acetyl-CoA if energy is not being used? Instead of entering the Krebs cycle, it won't anymore because marami ka ng ATP. Acetyl-CoA is instead used as a precursor for fat synthesis, triglyceride. But what if you need energy, but you don't have a source of carbohydrate? What would happen to acetyl-CoA? Acetyl-CoA will be used for the synthesis of ketone body. This occurs under the following condition: starvation, excessive fatty acid oxidation. And this is dangerous because ketone bodies can result to ketoacidosis. But we shall be focusing here on acetyl-CoA entering the Krebs cycle. So in this simplified diagram, acetyl-CoA, which is a 2-carbon molecule, combines with oxaloacetate to yield citrate. So oxaloacetate combines with acetyl-CoA to yield citrate. This starts the Krebs cycle. Citrate is converted into isocitrate, and then isocitrate is oxidized to alpha ketoglutarate. At the same time, it loses one carbon and release in the form of carbon dioxide. It also converts NAD to NADH. So now we have a 5-carbon molecule called alpha ketoglutarate that undergoes another oxidation step and also loses another carbon and release in the form of carbon dioxide. So what you have is succinyl-CoA, which is now composed of 4 carbon. This step right here is catalyzed by isocitrate dehydrogenase, whereas this step is catalyzed by alpha ketoglutarate dehydrogenase. The NADH that are generated here enters electron transfer chain. Succinyl-CoA is converted into succinate, and in the process, there is substrate level phosphorylation where GDP is converted into GTP that will be eventually converted into ATP. Succinate is oxidized to fumarate. That generates fat H2. Fumarate is converted into malate. And the final step is that malate is oxidized to oxaloacetate, generating the third NADH. So this now, again, restarts the Krebs cycle by combining with acetyl-CoA. Let's go into the details of the process. Okay, so pyruvate enters the mitochondria and is converted into acetyl-CoA. Acetyl-CoA comes from pyruvate, right? Acetyl-CoA enters the Krebs cycle by combining with oxaloacetate to form citrate. So this part comes from acetyl-CoA and this part comes from citrate. So there is a 6-carbon molecule. Citrate is isomerized to form isocitrate. Simple rearrangement. Isocitrate is decarboxylated. This carboxyl group is released in the form of carbon dioxide. So from a 6-carbon, it becomes a 5-carbon molecule. At the same time, isocitrate is oxidized to yield NADH and alpha ketoglutarate. So the enzyme here is isocitrate dehydrogenase, one of the regulatory enzymes in Krebs cycle. The 5-carbon molecule called alpha ketoglutarate undergoes another oxidative decarboxylation, wherein this carboxyl group is released in the form of carbon dioxide. So from a 5-carbon, it becomes a 4-carbon molecule. At the same time, this is oxidized to yield succinyl-CoA, dinagdagan ng CoA, generating NADH. The enzyme here is alpha ketoglutarate dehydrogenase. Now, this is the second regulatory enzyme in Krebs cycle. Succinyl-CoA is converted into succinate by releasing the CoAsh. And in the process, there is energy that is released and used for substrate level phosphorylation that generates GTP that will be converted into ATP later on. Succinate is oxidized to fumarate, catalyzed by the enzyme succinate dehydrogenase, and it generates FADH2, the only FADH2 that is synthesized here. Fumarate is converted into malate, and malate is finally oxidized to oxaloacetate. This generates the third NADH and catalyzed by malate dehydrogenase. And of course, oxaloacetate recombines again with the next acetyl-CoA to start the Krebs cycle all over again. So that would be Krebs cycle. If it's difficult for you to remember, this is a mnemonic that can help. So what are the reactants for Krebs cycle? The mnemonic is, can I keep selling sex for money? Oh, yes. So citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. But what about the enzyme? This is another mnemonic that you can use to remember the enzyme. So at another dance, Devon sipped down 5 drinks. Citrate synthetase, aconitase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, succinyl-CoA synthetase, succinyl-CoA dehydrogenase, fumarate, and malate dehydrogenase. Right, so what are the products of Krebs? Now, for every acetyl-CoA that enters the Krebs cycle, 3 NADH are generated, 1 fat H2, and 1 GTP. What about for every glucose? What would be the yield of Krebs cycle? Now remember that 1 glucose generates 2 acetyl-CoA, so everything has to be multiplied by 2. 6 NADH, 2 fat H2, and 2 GTP. And ultimately, all of them will be converted into ATP. So let's end Krebs by remembering these key points that you need to absolutely remember. Krebs is an amphibolic pathway that takes place within the mitochondrial matrix. What are the products of Krebs? 1 round generates 3 NADH, 1 fat H2, and 1 GTP. What about the enzyme? What enzymes are regulatory? What do you need to absolutely remember? There are 2. Isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. So this brings us to the final part of cellular respiration, and that would be the electron transport chain. All right, so the final stage of cellular respiration is the electron transport chain, where oxidative phosphorylation takes place. The ETC releases chemical energy that are stored in NADH and fat H2. How much ATP are generated for every reducing equivalent? For every NADH, 2.5 ATPs are generated, and for every fat H, 1.5. So I'll explain later on how we came up with this number. The electron transport chain is actually composed of a series of molecules embedded on the inner mitochondrial membrane. On the inner mitochondrial membrane, we have there complexes that make up the electron chain. For the sake of simplicity, we just represent them as complex 1, complex 2, complex 3, and complex 4. Complex 2 is represented here as coenzyme Q. Now, another enzyme that is linked to the electron transport chain is the enzyme ATP synthase, and this is the one responsible for the synthesis of ATP. Electron transport chain is responsible for a phenomenon called chemiosmosis. So what is this chemiosmosis? The electron transport chain pumps out hydrogen ion from the matrix into the intramembrane space. So the pumping out is called chemiosmosis. This results into in the charge, and we call it the chemiosmotic gradient. It is the chemiosmotic gradient that is responsible for the synthesis of ATP, as we shall see later on. So how does chemiosmosis and chemiosmotic gradient generated by the electron transport chain? So let's imagine this as the 4 complexes of the electron transport chain. embedded on the inner mitochondrial membrane. And let's imagine that they don't have any electrons yet. They are in the oxidized state. NADH1 that comes from the Krebs cycle transfers its electrons to complex 1. As it accepts electron, complex 1 pumps out 4 hydrogen ion coming from the intramembrane space into the mitochondrial matrix. This now sets the state. This process is what we call chemiosmosis, that sets the stage for the chemiosmotic gradient. The electron is transferred by coenzyme Q to complex 3, and as it accepts electrons, it pumps out 4 hydrogen ion from here into the intermembrane space. This is again chemiosmosis that now intensifies the chemiosmotic gradient. Electrons are transferred by cytochrome C into complex 4, which in turn pumps out 2 hydrogen ion, contributing to the chemiosmotic gradient. Now, for the complexes to continue running, the electrons has to be cleared out, and the final ultimate electron acceptor is oxygen. As it accepts electron, it combines with hydrogen to form water. Now, what would happen now to all of these protons that have been pumped out here? This is responsible for the chemiosmotic gradient. They would all want to go back in, but they can't because the inner mitochondrial membrane is impermeable. But there's only one way that they can go back in. This is by passing through a tunnel. This tunnel is formed by the enzyme ATP synthase. So they will now pass through the tunnel created by ATP synthase. And as they pass through, ATP synthase turns around like a turbine. And in doing so, it combines ADP and inorganic phosphate to form ATP. Now, as a matter of fact, for every ATP, 4 hydrogen ions are needed to pass through. Now, since NADH generates this much hydrogen in the intermembrane space, 1 NADH is equivalent to 2.5 ATP. This is 1 ATP, another ATP, and well, this is half ATP, but there's no such thing as half, so we just run this off to 3 ATPs. And what about FAD H2? FAD H2 bypasses complex 1 and instead passes its electrons directly to complex 2. And in the process, it generates a smaller amount of chemiosmotic gradient, but still nonetheless contributes to the chemiosmotic gradient. So FAD H2 is capable of generating 1.5 ATP. Now, I'm sure you are familiar with the poison cyanide. Cyanide is attracted to cytochrome iron of cytochrome oxidase found in complex 4. If cyanide binds to the complex 4, this now blocks the passage of electrons from complex 1 to complex 4. So all the complexes are locked in their reduced state, and they would not be able to accept new electron. This results in the failure to pump out hydrogen ions. So there is no chemiosmotic gradient. And if there is no chemiosmotic gradient, there would be no ATP synthesis. Since the passage of electron is also blocked, oxygen cannot be converted into water. So oxygen remains unused in the presence of a cyanide poison. Right? So just a summary, NADH generates 10 hydrogen ion, right? And this is equivalent to 2.5 ATP. Well, fat H, since it bypasses complex 1 and directly into complex 2, generates 6 hydrogen ions. That is equivalent to 1.5 ATP. So just to recap, on the inner mitochondrial membrane, we have the different complexes of the electron transport chain, the passage of electrons from complex 1, 2, 3, and 4 is actually a form of electricity. And that movement is used to pump out hydrogen ion from the mitochondrial matrix into the intermembrane space, the phenomenon called chemiosmosis that generates the chemiosmotic gradient. The chemiosmotic gradient is used to power ATP synthesis. When they go back in, ATP synthesis takes place by linking together ADP and inorganic phosphate to form ATP. The electrons are cleaved out from complex 4 so that the electron transport chain continues running. And the ultimate electron acceptor is oxygen, and it actually forms water. Okay, let's end today's review with these key points to remember. So the electron transport chain is the final stage for aerobic cellular respiration. The electron transport chain consists of a series of protein complexes that are embedded on the cristae of the mitochondria. The movement of electrons down the electron transport chain creates what we call the chemiosmotic gradient, and this gradient is what drives ATP synthase. NADH passes electrons to complex 1. This generates 2.5 molecules of ATP. Fat H2 bypasses complex 1, passes directly to complex 2. This generates 1.5 ATP. Electron transport chain requires oxygen to run because oxygen is the ultimate electron acceptor. Now, the poison cyanide is an inhibitor of complex 4. So that's the end of today's lengthy lecture.